Hello, hello. Welcome, everyone. Thanks for coming out. It is my great honor to introduce Dr. Dave Toews. Dave got his bachelor's at Acadia University and did a master's and Ph.D. at UBC, University of British Columbia, and has had two prestigious postdoctoral fellowships, one from NSERC and the other was a Banting, both Canadian government fellowships, and is now an assistant professor at Penn State University since 2019. And but other than that, I mean, Dave's very highly accomplished scientist, but he's also just like a great person and a great friend. And I think he studies hybridization of species in species, and I thought that was really interesting to think from a conservation point of view, because we rely on species being listed as endangered or threatened, but he asks the question, what is a species? And so without further ado, let me give it over to Dave.
Thanks, Toews. So I'm going to try and stay in front of the computer, but I'm probably just going to wander. So thank you so much, Toews, for inviting me. This has been great. I've been to Florida before, but never been to Archibald. So this has been amazing. It's been great chatting with folks about projects, not just Scrub J related work. So today I'm going to tell you a little bit about the research that we've been doing in my lab, as Toews mentioned, at Penn State. Currently, but research that was done before, and it is my job today to try and make some of the genetic and genomic tools kind of exciting and accessible for everyone here. So if I'm failing at my job, let me know. Please feel free to interrupt me. But before I begin, Toews asked me to tell you a little bit about myself and where I came from, which you covered a little bit. I grew up in a small town in Nova Scotia. This is east of Maine. A lot of times I tell Americans that I live from east of Maine. There's nothing east of Maine. So I did a bachelor's degree in Wolfville, Nova Scotia. Then I went to the other coast here in Vancouver, BC, where I did my master's. In Canada, it's much more common to do a master's before jumping into a PhD program. I actually worked for the essentially the Canadian version of the Fish and Wildlife Service in between here. I decided that, I didn't want a boss, and I liked doing research on more esoteric things. So I went back to PhD, and then I was very lucky to be able to have the opportunity to work at the Cornell Lab of Ornithology for almost 4.5 years. So they had to drag me out of there kicking and screaming, but I didn't go far. So I'm at Penn State here in Southern Pennsylvania. And it's been fantastic. Some of the hybrid zones that I will be talking about today between golden wing and blue wing warblers are literally 5 minutes outside my down the road. So it's a really wonderful place to be for bird diversity, but also salamander diversity and plant diversity. So it's been a really wonderful journey. But today I'm going to talk a lot about the rapid accumulation of species. So as evolutionary biologists and ecologists, we are really interested in understanding the origin and maintenance of diversity. Diversity can be measured in a lot of different ways, but one of the simplest is by simply counting the number of species. And a lot of our focal systems have been what are called adaptive radiations. And the radiation just refers to, a large number of species being produced in a very short period of time. So for those of you who have been to Hawaii and seen the Hawaiian honey creepers, this is, it used to be over 50 species. Unfortunately, there are a number of species that are now extinct. But it's thought that this diversity in form and function and color and, sort of all aspects of things that we value as biologists occurred over a very short period of time. It's thought that all of these Hawaiian honey creepers arose from a finch-like ancestor less than 10 million years. So this is a lot of species over a very short period of time. And so I have been studying in my own research a continental radiation of wood warblers. So these are otherwise known as the parulid wood warblers. They're a colorful group of North, Central, and South American species, songbirds. Many of them are migratory, and they are primarily insectivorous. And the variation that we see among wood warblers differs a little bit from sort of the classic radiations, like say the Hawaiian honey creepers or sort of the Darwin's finches, where we see a lot of dramatic differences in, say, bill shape and resource acquisition. My postdoc advisor has a moderately gruesome slide, depending on how you feel about a number of different species of warblers with all of, study skins, all of their feathers on, and then shows the skeletons of them. And it's very difficult to distinguish any of these species based on sort of those core morphological characteristics. And so we think this diversity, depending on what taxonomic authority, whether you lean towards more of a splitter or a lumper, we're talking about 110 species. And this diversity likely arose via the joint role of both natural and sexual selection. So differences arising from, evolution of different environments, but also, sexual selection based on different female preferences for different colors, traits, and songs. And so a lot of studies of evolutionary radiations have focused on those sort of, the morphological characteristics in Darwin's finches of, what are the selective pressures that has led to this diversity in bill shape and size. In this case, we've been interested in learning both about the evolution of color, but now, with the advent of genetic and genomic tools, actually trying to understand what genes might be responsible for this diversity. And so this is actually a review paper that was written in 2017, which inspired a lot of my thinking on this subject. These are actually, Berner and Salzberger actually work on cichlids, so another famous radiation in the Rift Lakes of East Africa. But they were interested in asking what we can learn and what we know about the genomics. What are the genes that underlie these spectacular radiations? And so we have some knowledge, of course, in Darwin's finches of some of the genes that are now responsible for this great variation in bill size and shape. This gene ALX1 may or may not be familiar to you, but as someone who went to the University of British Columbia, where they did a lot of stickleback research, I had been immersed in this world of sticklebacks. And a lot of research has been done on actually understanding the genetic basis for, in this case, variation in these lateral plates and also these spines that are presumably involved in defense and predation, defense against predation. These two genes, EDA and KIDX1. And then also in this another sort of charismatic evolutionary ecology system, heliconius butterflies throughout South and Central America, this gene optics has been shown to be involved in different coloration and pattern differences in butterflies. This is likely arisen by a mimicry, where if you are sort of matched to your local phenotype, you're less likely to be predated by birds. And so I was interested in, taking these types of questions and asking, what can we learn about the genetics underlying this diversity of a fairly common family of birds in North, Central, and South America? But before I can do that, the question here is, how do we go about connecting the genetic basis for any of these defining traits in this radiation of birds? This seems actually like a bit of a fishing expedition. So where do we even begin, with this non-model group of species? And so we have to have some knowledge of, say, candidate genes or some genetic basis for these differences to try and make these connections.
And so the great thing, yes, candidate genes are essentially just any gene that we think has some association with a phenotype. So it's a candidate for underlying that phenotype. Excellent question. So if we were working in one of these beautiful model systems, say sticklebacks or those heliconius butterflies, the reason why these are model systems is because we can bring them into the lab and do crosses. So we can cross fish with different plate numbers, butterflies with different coloration patterns, and we can use these genetic crosses to learn about connecting genes to the wonderful species defining traits. So to say, if I were to bring one of my warblers into the lab. So here I've got a red bird and a blue bird, and I want to understand the gene that's responsible for this color difference. I then do a cross. So I make an F1 generation. So I'm sure the mosquito fish people here are attuned to what we're doing here. But in this case, we're creating an F1 generation. So in this case, all of these birds are have one copy from, in this case, mom, this copy from dad. So they're all going to be, let's say, purple. This next generation, we take these purple birds and mate them amongst each other. And so this way, we can sort of recapitulate, we get this whole gradient of phenotypes going from red offspring all the way to blue offspring and everything in between. And I'm not going to go into the details of sort of how this process works, but it has to do with this wonderful process of sexually reproducing organisms called recombination, where we're mixing up genomes of these birds, and presumably we're mixing up the traits that underlie, in this case, this coloration difference. And so then if I go out with some gene sequencing technology, I go out and sequence their genomes, I can then try to find a little spot on their genomes that correlates with this color. So going, in this case, we would say, okay, this is a part of the genome that associates with this coloration difference going from red and blue. And so, yeah, we would have a candidate gene like ALX1 in Darwin's finches that we could go out and survey and understand better. Unfortunately, yes.
Are you in the F1 generation, it's F2, are you crossing the
Yes, so you're crossing F1s with F1s to get F2s. Yep. There are, fruit fly people have all sorts of different tips and tricks that they can use, but in this case, in most cases, you would just be crossing F1s. However, as you're probably realizing by now, I cannot bring neotropical migratory insectivorous wood warblers into the lab and successfully create these types of crosses. And so what we do to get around this is not use what we would say like a laboratory setting, but we take advantage of natural hybrid zones. And so these don't obviously occur between all species, but in some cases where we have the range, in this case of these red birds, that comes and maybe abuts the range of these blue birds, that there might be an area in between where we actually get naturally formed hybrids. And so this is, I'm showing a little schematic on the bottom here, where we're sort of running a transect, a physical transect between the range of species A and species B, and we have the frequency of some trait that we're measuring, in this case, coloration, or some genetic marker. And we see this transition between the two, and this is where all these hybrids are formed. But now nature's kind of done the hard work for me. I didn't have to create these hybrid warblers in a lab. I can then take this natural variation in the wild, use that type of gene, some sort of gene sequencing technology. Now we're doing whole genome sequencing and try and connect these coloration characteristics with a certain part of their genome. And so this is kind of exactly what we have done over the past few years, studying hybrid zones between closely related species and trying to connect these species defining traits. And like I said, not all species form hybrid zones, at least in North America. We think a lot of them arose through different, through isolation into different ice-free refuges during the last glaciation. So this is a sort of what North America looked like during the height of the last glaciation. You can see Pennsylvania is actually in a pretty cool spot. We were sort of right in the middle of this mile thick of ice. It kind of blows my undergrad's mind sometimes when I tell them, in fact, how much ice there was here. But in this case, we have a lot of evidence and instances where species that were once sort of connected throughout, say, that lived happily in the boreal forest were split into different ice-free refuges in the southeast versus the southwest of North America. That then, after these glaciers receded over many thousands of generations, they came back into contact, so they expanded their range further to the north, and then eventually came back into contact. So we've got lots of examples of east meeting west, where you have, at least in the Great Plains, I've done a lot of work along the eastern foothills of the Rocky Mountains, and then also areas here in the Northeast, which I'll talk about, which were thought to be these areas where these contact zones, hybrid zones came about when you had these isolated populations that eventually came back into contact. And so how did we go about doing this in wood warblers? So what I'm showing you, please, you will lose an eyeball if you try and read the names here, but this is what was the best phylogeny of all 110 species of wood warblers done by my postdoc advisor, Irby Lovett at Cornell. So this is showing all the different genera and all the different species across all these wood warblers. And what I'm going to do today is start by telling you about a single hybrid zone that we used to try and connect genotype or the genes underlying these specific phenotypes. So I'm going to zoom in for part of this talk, and this one little itty bitty tip right up here, which is made up of 3 species. These are warblers. These are golden wing and blue wing warblers. And unfortunately, the other species in this genus is called Bachman's warbler, one of the only North American songbirds to go extinct. We actually have a paper on its genome that's coming out soon. I will not talk about that today. But what I am going to do is tell you a little bit about this mystery of golden wing and blue wing warblers. And I wasn't the first to be perplexed by this. But even those of you who aren't familiar with birds or warblers or anything can probably tell that these 2 species look pretty distinct. There are actually barely 2 feathers on them that are the same color. So phenotypically, in terms of their coloration, they look very different. They also sing quite different songs. However, in a letter in the early to mid 1800s, John James Audubon wrote to his friend Bachman about the various different species he was seeing in North America. And he listed golden wing and blue wing warblers, but then he put same species ? That was odd. They seemed quite distinct based on, all the characteristics that I just talked about. In fact, the plot thickens a little bit at the turn of the 20th century when this 3rd species, which was called Brewster's warbler, was suggested to not even be its own species whatsoever, but was in fact suggested to be a hybrid between golden wing and blue wing warblers. Now, Audubon didn't know that golden wing and blue wing warblers hybridize necessarily. And this is a paper, this is a kind of a classic in my like super niche field. But it was, just this guy, Walter Faxon, were studying this pair that he observed of golden wing and blue wing warblers that were nesting in the swamp in his backyard, and found that they created what was at the time described as an entirely distinct species. And so it was quite heretical at the time, but it was sort of careful observation that he demonstrated that in fact this was a hybrid between golden wing and blue wing warblers. In fact, they only occurred where both of these species also co-occurred. And we now know with the abundance of natural history collections that there's a wide range of hybrid phenotypes. So these are what we call the sort of parental plumage types. So golden wing and blue wing warblers. Golden wing warblers have this really distinctive black throat patch and black mask, which is lacking in blue wing warblers. And then you see, this is what would have been called a Brewster's warbler before, but then this was also called its own species. This was called the Lawrence's warbler. So these different hybrid combinations were so different from the two parental taxa that they were thought that they were their own species. And there's a very interesting but dynamic nature of hybridization between these two species. So here I'm showing you the ranges. This is, in goldenrod is the range of the golden wing warbler. In blue, the range of the blue wing warbler. And then these areas in baby blue, which illustrate areas of overlap where we find lots of hybridization. So this is where kind of I am here in central Pennsylvania. We get sort of more, actually we get like a pretty even mix these days. They like early successional habitat. So kind of like the scrub jays, I like to say these are very particular about the crappy habitat that they like. So, power cut lines are like pièce de résistance to golden wing and blue wing warblers. But because they like a type of habitat that's very dynamic on the landscape, it means the hybridization dynamics has actually been quite dynamic between them. And so folks have been interested in particular because certain populations of golden wing warbler, especially throughout Appalachia, have been declining, in some cases drastically, like 95% declines throughout these populations. The main range, what we call the main range, is doing a bit better. But in many populations, like where I was living in Ithaca, New York, people studied golden wing warblers throughout the 40s and 50s. You cannot find a golden wing warbler in Ithaca, New York anymore. It is only blue wing warblers. So there's this weird pattern of replacement, there's patterns of declines, and there's this very dynamic situation of hybridization. So given all this, folks had been interested in developing genetic markers to try and distinguish these two species. But there were some problems with that. So, given that they are so phenotypically distinct, they are described as distinct species, one would think that finding, parts of their genome that differ would be relatively straightforward. However, previous researchers using different types of nuclear genetic markers, they're looking for what we call fixed differences. These are sort of species-specific, unique differences. And so we would expect distinct species to have many of these fixed differences. And so the details of these different types of genetic markers don't really matter. What matters is that given, even greater resolution throughout the genomes of these birds, there were still no fixed differences. Nothing that was specifically golden wing-like, nothing specifically blue wing-like. And so when I started working on the system, my colleague Scott Taylor and I, we developed what we thought was like the latest and greatest, 16,000 of what are called these rad markers. Again, it doesn't matter, but we're looking at 16,000 parts of the genome. These have been used to distinguish individual populations of the same species. No fixed differences. So we had just spent a lot of time and a lot of money. My postdoc advisor was not super impressed. But we had just started riding this wave of genomics and whole genome sequencing. And so we thought, okay, let's just do it right. Screw just looking at little snapshots throughout their genome. Let's actually sequence the whole thing to try and find the bits, just sort of the needle in the genomic haystack that makes a golden wing warbler a golden wing warbler and a blue wing warbler a blue wing warbler. Because again, these are two birds that we caught in the same net. They look so different. There were lots of interesting theories that were like, well, okay, if there's not a genetic difference between them, maybe it's an environmental cue that they, were raised on some different diet. And so they express some different hormone. And so there was anyway, there were lots of interesting theories, but the easiest was always going to be whether there was a genetic difference. So we started this study by sort of sequencing 10 whole genomes of golden wing warblers and 10 whole genomes of blue wing warblers, kind of spanning what we would call the phenotypic spectrum. So birds that looked like good parental species. We've now followed up, we've sequenced hundreds of genomes of both of these species. The story does not change. So just to give you a little bit of a flavor for some of the field work that we do, this is one of our 9-meter mist nets. And so to catch these birds, we go out into their territories, we set up a song playback, and they're not, you could either consider them very smart or not very smart, but they'll usually come down, investigate the speaker, and if we've set up our mist net in the correct spot, they will fly into it. And like the scrub jays, actually, this is a pretty sweet habitat to work in terms of mist netting because stuff is pretty low. Although you guys have like, pre-made mist net lanes, which is pretty sweet. So we have to make our own, but machete doesn't cost that much money. And so we take all sorts of field measurements. We take 1 million photographs, but most importantly, we take a blood sample. And working in genomics on birds is, complicated for lots of reasons because I can't raise them in a lab, for example, but they have nucleated red blood cells. So that might sound like lame and esoteric to those of you in the audience, but our red blood cells are not nucleated, right? They look like little kind of hollow inner tubes. Well, not hollow, but they don't have a nucleus. And so every single drop of bird blood just has an abundance of material to work with. So we, have blood samples. Well, I guess this won't impress this audience, but we have blood samples from the 90s that still works. But yeah, so there's scrub jay blood that goes back even further. Anyway, so we do standard measurements, photographs, and take a blood sample for genome sequencing. These are the individual birds that we went ahead and sequenced their genomes. You can see these are, what look like very good blue wings. These are what look like very good golden wings. And I am going to skip essentially all of the methods for how we went about generating this data and analyzing it, although I would be happy to talk about any sort of methods and approaches if folks are interested. But again, what we were looking for is what we called these fixed differences, these sort of species defining regions of their genome. And we measure this with a statistic. The statistic is called FST. So it's called the fixation statistic, right? And so it goes from 0 to 1. And for each sort of part of the genome, in comparing golden wing and blue wing warblers, 0 means that there's no difference between golden wing and blue wing warblers. They're essentially the same. They have the same DNA sequence. They have the same genetic marker at that position. By contrast, a value of 1 indicates a fixed difference, that golden wing warblers have a golden wing variant, blue wing warblers have a blue wing variant. And so here I'm showing you a histogram of these values across the entire genomes, of little windows across the entire genomes of golden wing and blue wing warblers. And what you can see here is that the average is basically 0. So this made sense why everyone else that was trying with lower resolution genetic markers to try and find individual differences basically came up with 0. But if you zoom in to this little tail that you can't even see when looking at the full histogram here, if you zoom into this little tail over here, you start to see a small number of genetic positions that look like they are fixed in the genomes. And so what we can do is then put this value on its side. And so what I'm going to show you is what's called a Manhattan plot. Is that, have I gone over time? Is that, oh, okay. No. Oh, the ornithological conferences now use like bird songs for like okay, you've got 5 minutes left, 3 minutes left, but it means that you're like scarred by listening to these like, every time I hear a chestnut-sided chickadee, I'm just like, oh, I've only got 3 minutes left. Okay, so it's, oh yeah, and so like we're talking now, we've gone up to 11 million positions in the genome. This FST is indistinguishable from 0, essentially 0.006. Like we have no statistical ability to say whether it's different from 0. But if we go ahead and put these on their side and look at what's called a Manhattan plot. It's called a Manhattan plot because you're looking for sort of the peaks in the sort of skyline of New York City. So you're interested in finding these buildings that the, Lady Liberty that stands out against a background. And so what this is showing you is the same data, but now I'm looking across the entire genomes, comparing, in this case, golden wing and blue wing warblers. And each one of these positions is a spot on a single chromosome in their genome. And what you can see are these 6 peaks, these 6 regions that stand out against an otherwise fairly homogeneous background. What is wonderful, well, what was wonderful? What was exciting for us is that, these regions are few, so there's only 6, and they're all very small in that, the largest is 300,000 base pairs. Now that might sound like a lot, but in the context of an avian genome is 1.2 billion base pairs. So these all of these together make up less than 0.03% of the entire genome. Our smallest is about 30,000 base pairs. And this size means that its location in the genome is very informative to what genes it's nearby. And in fact, most of these regions that were divergent between golden wing and blue wing warblers were right basically within or next to genes with some connection to the pigmentation process. So I'm going to highlight 2 today. The first is called the agouti signaling protein, also called ACIP. And the second is called beta-carotene oxygenase 2, otherwise known as BCO2. And so, at least in the case of this agouti signaling protein, it has a well-characterized function in the melanogenesis pathway. So birds like you and I, and the diversity of humans, color their integument or skin or feathers, in the case of birds, with melanins. And so agouti has been shown to be involved in melanin differences. These are the Monarchus flycatchers. A colleague, Al Uwe, looks at these melanic forms and has shown that this same gene, this ACIP agouti gene, has been involved in generating these melanic forms. Work in Japanese quails has shown that ACIP, the expression of this gene, is involved in the reduction or addition of melanin to these feather pigments. And so agouti, and even beyond birds, it's been shown in horses, cats, Parameciscus mice. It's become famous for being involved in the melanogenesis pathway, which was very cool. And we've actually demonstrated that it's strongly associated with this variation in throat and mask coloration. So if you have 2 copies of the golden winged agouti allele, that means you have a black throat and a black face mask. If you have a single copy of the blue winged allele, that means you no longer have a throat or mask. And that was the story for 99.99% of our birds, because birds either had, it was sort of on or off. You always had a mask if you had a throat, a black throat, and you always had a black face mask, or if you lacked it, you lacked it. Except this was a purely pandemic project. This was a bird that we found 15 minutes from my house. It was the first golden wing warbler that we found that had a really clear face mask, but lacked the entire black throat. And we found that it was this region of the genome upstream of this agouti gene. One part of that region controls the throat coloration, and one part controls the mask coloration. Given they're so close together in the genome, they're 99.9% of the time inherited together, except in the very rare instance when you have an instance of what we would call recombination in this hybrid, where this is a bird that lacked its throat but had the face mask. And so, looking at all of these individuals, we've shown, a very strong connection between a gene, in this case, agouti signaling protein, and this phenotype, this black face mask and black throat coloration. but this was already a gene that had, some previous connection to melanin and melanogenesis. But birds don't just color their feathers with melanin, they also use carotenoids. And so that's actually one of the most celebrated characteristics of wood warblers. They're kind of orange and yellow. You and I also use carotenoids, but we don't deposit it in our skin for coloration. Whereas birds deposit it in their feathers, and that gives them these wonderful, colorful hues of yellow, orange, and red. But like us, birds can't synthesize carotenoids all on their own. They have to acquire it from their diet. And actually much less is known about the biochemical pathways that give rise to variation in carotenoid characteristics. And so this was actually one of the first studies to document a potential candidate gene for carotenoid synthesis in birds. Well, lo and behold, but a few years later, on the cover of Science, these colleagues, Melgarag de Gazga, showed that this is in canaries. So this is a situation where they can do crosses and they can look at pedigrees and such. But the candidate gene for differences in this case in dichromatism between male and female mosaic canaries, which differ in the extent of a carotenoid-based red face coloration, was this gene BCO2, beta-carotene oxygenase. And so it's now been demonstrated. It's interesting, like the canary folks are able to do their crosses and demonstrate really strong gene and expression and phenotype differences, but they don't have sort of the relevance. If they weren't able to point to a study that actually showed this gene BCO2 being relevant in some natural population, you could say, oh well, that was just this is just because of domestication. So it's been interesting to see these different research programs go hand in hand. But okay, so now we have a couple of genes to go fishing with, right? So we have a couple of these candidate genes. There's others, but I'm just going to focus on these two for this specific talk. We have this agouti signaling protein, this ACEP, which is involved in melanogenesis. And we have this BCO2, beta-carotene oxygenase, which is involved presumably in carotenoid degradation. And so I want to motivate the next part of my talk by asking, okay, well, what exactly are we going fishing for? Why do we really care about having these candidate genes? To ask about their variation in this radiation. And then we're gonna answer this question, where are we going fishing? So the first question is, what are we going fishing for? And what we're going fishing for is evidence for what we call introgression. So I told you about these hybrid zones where you get, species 1 coming into contact with species 2, and they hybridize. That doesn't necessarily mean that there are genes moving from species 1 into species 2. If you have a case like the mule, for example, that's a dead end, right? They are sterile, they are not back crossing. If, however, there is back crossing, if you have hybrids mating with a parental type, you can have genes moving from one species into another. And we use this term introgression to refer to this process. And so why would this be helpful? Okay, so I'm a species, and I'm getting very tautological, but I am waiting for a new mutation that will give me some new fancy coloration pattern. Well, natural selection works by trial and error. These individual mutations that might, change a small part of a gene product one way or another way, these are, individual changes that may result in a species or a lineage becoming more fit to their environment over time. But as you can expect, mutations, the process is slow and it takes a long time to wait around for these pesky new mutations. why reinvent the wheel when your friend next door has already come up with the solution? And so this is the idea behind hybridization and introgression. So this is a little schematic where you have these two lineages, in this case represented by these little blue chromosomes and red chromosomes. These are, you know, if you look in the background, you can see sort of like a bifurcating tree. These are lineages that are kind of on their own trajectory. But in this case, you have an instance of hybridization. You have this one individual that is half blue, half red. It's a hybrid. That hybrid then went to reproduce with one of these blue individuals. And now their offspring are all mixed up. And so you have the introduction of these, in this case, red genes into the background of this blue lineage. And so you can have a situation where this red lineage has, say, you know, solved some evolutionary problem, has evolved to be better adapted to its environment. And through the process of hybridization and introgression, this blue lineage didn't have to wait around for all of those new little individual mutations. It could just borrow from its friend next door. And so we call this introgression. And I'm going to use a new analogy. This may be overly ham-handed, but we'll give it a try and see how we do. But the idea here is that, say you and your neighbor have the same model of house built in the same year on the same road, and your neighbor wants to put in a new front porch. And so they go through all the trouble of planning, making the blueprints, getting a contractor to put on this front porch. So here we have your house and your neighbor's house. They're basically the same, but your neighbor just went through all this process, got Mike the builder to come by and put on this beautiful new front porch. You could go ahead and reinvent the wheel, spending all this time going through the planning process, getting a new contractor, or you could just borrow their blueprint, borrow their contractor, save yourself a lot of time, and in this case, a lot of money, and put yourself in a place where you get a fancy new front porch. This is analogous to this process of introgression. And you may think, well, Dave, why is this relevant? You know, has this ever happened? It's happened and it's been documented in lots of different systems, even our own. So I think one of my favorite examples when I'm talking about this is evidence of what we call adaptive introgression. So that takes this idea of gene movement from one group to another, but saying that it's adaptive, that it's actually beneficial, that it's, you know, increasing in fitness those individuals that have that gene that was beneficial. So this is an example from these Denisovans. So this is a lineage of hominid that's only known by a few finger bones, but they, through the magic of genome sequencing, have, you know, obtained whole genomes from these Denisovans and found that the genes retained from Denisovan ancestry is non-random. Specifically, in Tibetans living at high elevation, they have a different variant of a gene called EPAS1. Doesn't matter what the gene is, but in this case, it's involved in blunting the hypoxic response for individuals at high elevation. And when you look at the whole universe of different EPAS1 gene variants, Tibetans have a very unique, they've got a very different set of DNA mutations there. And they all associate with those two finger bones that were found in those Denisovian individuals. So this is a case where the interpretation of, I mean, these seem like discordant patterns, but through this process of adaptive introgression, it's likely that, our hominid ancestors were known for evidence of hybridization. And in this case, introgression from Denisovans into ancestral hominids. And it's been retained because it is beneficial at those high elevation populations. So we're going fishing for evidence of introgression. And now, where are we going fishing? So obviously we're going throughout all of our parulid warblers. So we have our candidate genes that we identified from our little tips over here, these golden wing warblers and blue wing warblers. And Up until a few years ago, we worked on the largest genus in this group. This is the Pseudophaga genus. I will tell you now, we have whole genomes from the whole radiation, but we haven't analyzed all of this data yet. So this is the story that I will be telling today. But we have found these two genes, agouti and BCO2, that are associated with these phenotypes in golden wing and blue wing warblers. Well, we see these same phenotypes, the black throat, the black mask, as well as variation in the distribution of carotenoids that vary across this other genus of warblers. So we're going to ask, okay, do we see evidence of introgression across any of these species of Pseudophaga warblers? So this is about 35 species. And I'm just going to show you, this is the data set that we generated. So this is a phylogeny of these Pseudophaga warblers, and each one of these little lollipops represents a whole genome sequence. So our goal here was to have multiple individuals of every species. Some of these were hard-fought samples. Many of them were loaned from museums. Some of these species occur only in small populations or of conservation concern on Caribbean islands. But we have, in this case, over about 36 species and over 150 samples of whole genomes. And so how are we going to go about analyzing this data? So I'm only going to show you one method, so don't worry, I won't go into the methods too far. But in this case, we're going to do what is called a species tree gene tree comparison. So in this case, this is what we call a species tree of these four hypothetical bird species, A, B, C, and D. And these gray outlines and these colors illustrate the actual relationships among these species. So B and C are more closely related to each other than A and C and A and D. So this is what we believe to be the true representation of how these species are related to one another. By contrast, on the right, I'm showing what's called a gene tree. And so this is a specific portion of the genome that's reconstructing, say, the relationships at a specific region, say agouti or BCO2. In this case, this is the relationships from our species tree, right? This is what we believe to be the true answer to how related these species are to one another. But we see in this case a slightly different pattern. So in this case, we have the green species, C, more closely related to D. How might this come about? There are a number of different mechanisms that can produce this, but the main one that we're going to focus on is this process of gene flow and introgression. And you can imagine, if you have a gene going from, say, species D into species C, they're going to look more similar to one another than other parts of the genome. So this is what we're going to do. We're going to compare what we believe to be the good species tree, and then these gene trees for these two candidate regions. So here I'm showing, don't worry about the names or even the orientation here. This is an example of a species tree. So this is all the Pseudophaga warblers plus a couple outgroups. What we believe to be the best representation of their true relationships. On the right, I'm now showing just a tree built with sequences generated from that agouti pigmentation gene. And what this is showing, this is called a co-phylogeny. So we've got the same species mirrored on the left as mirrored on the right, and these red lines trace their connection. So here we've got Magnolia and Magnolia. When you see a nice ladder, so you see straight lines up and down this, what's called a co-phylogeny, that suggests that you have the same pattern from your species tree as you do from your gene tree. And so this is kind of consistent, that they're not telling you a very different story. So in fact, this suggests that, aside from a couple, squiggly lines here, these are actually species that don't have as high a confidence. This suggests that, you have a pretty strong mirrored pattern, that agouti, this acyp tree, generally mirrors evolution of the genus more broadly. However, the same pattern, the same approach with this different gene, BCO2, tells us a very different pattern. So in this case, we've got many more crossed wires. We have, in this case down here, we have a lot of species that look more closely related to one another at just this small sliver of their genome, including this BCO2 gene, than what we believe to be the true branching pattern. And I want to highlight, so I'm going to erase most of these because I'm just going to focus on this one group. So these are 4 species down here. This is otherwise known as the Blackburnian warbler, the yellow warbler, the magnolia warbler, and the American redstart. For those of you who are familiar with North American warblers, they're pretty common North American species. But these are otherwise, not closely related species in terms of their actual branching patterns, in terms of how we think their relationship, how they're related to one another. But by contrast, their BCO2 sequence is almost identical. And in fact, they share what is, a common amino acid substitution. So I haven't gone too much into the detail of the actual gene sequence, but gene sequence only tells us one thing, right? We actually are interested in the protein that is produced by that gene, which creates this enzyme. And these all share a single amino acid change that is otherwise conserved across all bird genomes that have been sequenced, which suggests that there might be something interesting and functional about that change. They all share those and several other mutations. And so we call these guys the carotenoid crew, because they're also well known in this radiation for having a really broad distribution of carotenoids. So yellow feathers, yellow and orange throughout their bodies. And, I've only shown you this, what's called this co-phylogeny. We've done other explicit tests to show that this discordance between this tree and this tree is actually due to this process of introgression, that this similarity is due to the fact that there was hybridization at some point in their past. So just to show you as this little schematic, so here we have, in this case, this is a redstart, our Blackburnian warbler, our magnolia warbler, and this is our drab proto-ancestral yellow warbler. Now, yellow warbler is obviously named for the fact that they are so yellow, they have so many carotenoids. But in this case, in this schematic, we have, this BCO2 variant that evolved across these species. Maybe they were, moving this gene back and forth with one another, but then at some point in the past, this ancestral yellow warbler had some hybridization, say, with this magnolia warbler. There was introgression of this maybe more colorful BCO2 variant, and that's what gave us this beautiful, colorful yellow warbler. This is, we don't know about which species were necessarily involved, what the directionality was. These are really hard questions to get at. But we think there were at least 2 bouts of introgression among these species sharing this one gene region that we know, has a functional consequence for carotenoid deposition. And actually, if you bear with me one more second, as if we zoom in on one little spot of this BCO2 gene region, that spot that includes that little amino acid substitution that I was just talking about that might be functional. If we do another phylogenetic tree from just this one little sliver of this BCO2 gene, we actually get 2 more species that pop out. This is the prairie warbler and its closely related Caribbean island endemic, the Vitulins warbler. These are also species with a very broad distribution of carotenoids across their body. And so, this has been quite exciting. We've been able to go from, these candidate genes underlying these, really dramatic plumage differences to actually demonstrating that there's, not only a gene to plumage coloration or gene to phenotype connection, but actually that there's been evidence of introgression and gene flow amongst these different species. So up until a month ago, this is where this talk ended. However, over the past couple of years, we got a big grant from the National Science Foundation, and we've affectionately called it the Perulome Project. So I don't know, the penguin genome people got to call theirs the penguinome, and that got a lot of traction. So I'm giving it a shot. I don't know. We'll see if it works. But I showed you this project from all of these genome sequences from all of the largest radiation. Well, we now have whole genome sequence from every single warbler species in the entire radiation, usually represented by at least 5 individuals. So this is data that's just recently been collected. This is primarily samples loaned from a really amazing and diverse group of museums throughout North America. But I was interested in asking the lowest hanging fruit. Can I just pull out this beta carotene oxygenase sequence from all of these species? Do we see any other species beyond just this single genus that looks like it has been involved in this introgression? And so you're not allowed to talk about this beyond this room, but this is now the phylogeny of all warblers. So now, okay, I can't even represent all of these species legibly on a single sort of up and down phylogeny. So now this phylogeny is basically in a circle where this is the common ancestor. So the root here, and all of the species are represented in a circle. Again, you don't have to worry about the names, but what we found, so this is just a phylogenetic tree of this one little sliver of BCO2 that we found was introgressed among these different species. So here's our carotenoid crew here, and we have one new player here, and this is the Wilson's warbler. So again, this is another common North American species with broad distribution of body carotenoids. We also found some other interesting amino acid substitutions at this one site that we think is functional and important. And this is, this is the amino acid substitutions we found in these, the carotenoid crew. And this is a new one we found for this, these are like Nashville warblers and Virginia's warblers, et cetera, for those who are into the warbler world. But the other one that I was super excited about is the only other species that shows an amino acid substitution is prothonotary warbler, which, beyond the yellow warbler is just the lemoniest lemon yellow warbler out there. Some people try to convince me to try and say it prothonotary. I don't know. Okay, I'll just stay with prothonotary. So this is, we're literally just scratching the surface of this data set. There's a postdoc that's coming on this summer to work with me in our lab to try and analyze, beyond just these. I've been cherry picking and focusing on these candidate genes that we have some intuitive connection to their phenotype, but we have whole genomes that we're going to be working with. We think, at least with these North American species, given the dynamic nature of their evolutionary history, that their ranges, contracted, receded, they came back into contact. They had lots of opportunity for contact, for hybrid zones, and for there to be introgression. And, as a side note, I've got a small cottage industry of like identifying using genomic tools, these weird intergeneric hybrids. And so all these are, these are absolutely fun, like little ornithological mysteries, which get a lot of press more than my other stuff. But they showed the fact that these species retain, this is a hybrid between cerulean warbler in the Setophaga genus and a blue-winged warbler in the Vermivora genus. These are different genera that are able to produce an adult hybrid that sings and looks like a normal warbler. We don't know if it was sterile. We don't know if it reproduced. These things are hard to follow, but it presents both the opportunity And, the feasibility for these types of, deep scale introgression events, hybridization events, to have happened in the evolutionary history of these birds. And so, my hope is that adding to this, storied history of, being able to connect genes to phenotypes in these, species radiations, that we could potentially one day put these carotenoid processing genes and melanogenesis genes up there, and to eventually one day ask whether variation of these genes or their propensity for gene movement and hybridization has actually contributed to the diversity that we see. And that's going to be sort of a longer, larger question. So just to sum up, really our key foundation for us to even start working on this project was using these natural hybrid zones to identify these gene to phenotype associations. So I told you about these golden wing and blue wing warblers that differ at only a handful of their genome, and that allowed us to connect the actual genes that underlie those differences. I told you about, zooming out to the genus as a whole, that this gene, this carotenoid processing gene, BCO2, illustrates what we think are multiple bouts of gene flow and introgression in the history of this, this family. And we think that this might include a functional amino acid change. And I'm actually working with colleagues to follow up on this functional change in the lab. And then finally, we're just kind of getting started with this Perulum project where we've, been able to gather sequence from across this entire family. And I'm excited about all the various discoveries that are coming. So with that, I would like, thank you so much for inviting me to come and speak today. This has been an amazing opportunity to learn about what's going on here. And I'm just happy to be able to share some of the work that we've been doing. And I'm happy to take any questions. So thank you. Yeah. For vermivora, for golden wing and blue wing warblers, I mean, it's always, I love that question. I was telling someone today, the thing I love about this. So this is the Manhattan plot that shows the genetic differences between golden wing and blue wing warblers. And the same people look, or different people look at the same data and say, oh my God, their genomes are so homogeneous, they cannot be the same species. So they must be the same species. On the flip side, people look at this and say, aha, we found the 6 speciation genes that differentiate golden wing and blue wing warblers. My feeling is that beyond the genomic data, just based on the fact that they hybridize rampantly, there's no evidence of hybrid dysfunction, suggested out of the gates without even any genetic data that they act like different sort of plumage types of the same species. That said, I want to answer this question. And so the plan is going forward, because one of the problems with this is that plumage phenotype isn't a super great predictor of like genetic ancestry, but there's a ton of extra pair fraternity. And so we can't really figure out hybrid fitness because we don't know who the father really is. And so pairing this stuff with a really good pedigree population is our plan over the next few years. But so I successfully walked around the question. I'm gonna go in the back. Yeah.
On these 2 species, there was that guy in the 1800s who wrote that they might be the same species.
John James Audubon. Yeah.
Yes, how in the world he would have come to that conclusion?
So there's like 3 pages of that facts in the paper that's dedicated to just that one, because they do look different, they do sound different. It's unclear whether he had any access to hybrid specimens. And so there is the possibility of a misidentified specimen that he did look at, but it's still highly speculative. So I unfortunately don't have a good answer for you, other than to say the person that brought that to light also struggled with, whether he was a superhuman it's a great paper, or that they were missing something. So I don't have a great answer for that, but it's a very good question. Fair enough.
Could you repeat the questions for the?
Oh, yeah, sorry. I said I was going to do that, and obviously I blanked. Can I do that for the ones going forward? Okay, cool.
Okay, for your new project, is it just all current species, or are there any extinct species, since you said that they do like flexible?
Yeah, so the question was whether it's just extant species or whether there's extinct species as well. So yeah, so that Bachman's warbler, we now have whole genomes from 7 birds. I mean, that's a whole talk in and of itself, but it actually looked like, so Bachman's warblers look like one of the hybrid phenotypes. And so there was some suspicion that it also could have been a hybrid between golden wing and blue wing warblers. The answer is definitively no, it wasn't. It actually looked like it was a real thing. They shared a common ancestor maybe 3 million years ago. It's highly divergent. It shows some evolution at agouti, none at BCO2. And so it unfortunately really was a species that looks like it went extinct. It also looks like we also quantified runs of homozygosity. So and it had, which is an indicator of inbreeding. So it did have very high evidence of inbreeding, and so did golden wing warblers. But yeah, that's the only extinct species that I know about that would be represented in natural history collections, at least. But that's it's a great question. Yeah.
So jumping kind of briefly back to the first question, you said that in both of these species, there's really high extra pair fraternities.
Yes.
I was wondering how you would get a really strong pedigree.
So the question was, how would you get a strong pedigree if there's extra pair paternity? You have to sample all the males in the population. And so, well, not all the males, but like a good enough proportion. And then because, juveniles are going to disperse, I don't know if we have to do it. We have to do it because this has just been a question. So there's Fort Drum actually has the best concentration of birds here. Where's this is actually data from Fort Drum. And so this is like, you can actually see the change from 2008 to 2015. So we've got to do it soon because golden wings are going down, but there's like a really nice mix of both species plus hybrids. And so I just think it is a feasible operation with enough people dedicated to finding nests and banding as many birds as possible. But we have to do it at some point because otherwise, yeah. Yeah.
I was also wondering, speaking of the population declining golden wings, I wondered if you knew why that replacement seems to be occurring.
So I think it's so the replacement of blue wings to golden wings happens in certain populations, but not others. So the Appalachian group, that is the one that's tanking the most. And we don't see as much replacement there. And we think that decline has more to do with the fact that they have a different migratory pattern. They've got a different wintering area that seems to be getting hit harder by habitat alteration on the wintering ground than the breeding ground. Whereas this main range population, there are instances where there is, like I talked about in New York, where there is replacement. There was big concern in Southern Ontario that, there were lots of hybrids and that blue wings were going to move in. That was a concern back in 2007, and it doesn't seem to have come to fruition. So I don't really have a good answer for why there seems to be really strong turnover in some populations, but not in the other. It's partially just due to the genetics, right? The golden wing phenotype is the recessive phenotype. But yeah, we don't really have a great understanding of that. Yes.
Yeah, so in the examples you gave, like helicomias and sticklebacks, those are pretty classic cases of, as well, predation driving the selection pressure, which leads to a lot of adaptive introgression that you see. So I'm wondering, what is the selective mechanism you think is driving this actual color introgression? And is it like sexual selection, and if so, to do with sex chromosomes?
Yeah. So the question is, what do we think is like the evolutionary mechanism for driving this? So I get wishy-washy and just say the joint role of natural and sexual selection. Because I think there's folks that are interested in how the light environment might transmit some of these colors. So there might be a more direct and intuitive connection to the environment than we are, say, are articulating here. You know, there have not been a lot of direct manipulations, although the direct manipulations that have been done have been in golden wing warblers. So they actually bleached some of their little masks and their little bibs to make them look white. And so they had like they had lower fitness, but like they also bleached them. So it was like, there's some work in common yellow threads that look at the extent and size of the bib. That does seem to be associated with some fitness characteristics, but it's complicated because it varies by population. So, you know, I would, yeah, I don't have a great answer other than, you know, it's very possible. It's very possible both, but it's really, really unclear. I mean, like it is put forward as a classic example of sexual selection. But I also think the morphological similarity belies some of the ecological differences. So we've got a whole other train in the lab where we're doing fecal metabarcoding. So we're using genomic tools to learn about their diet, also learn about their microbiome. So a postdoc, Marcela Baez, she's been looking at microbiome differences. So using the warblers and the hybrid zones themselves as the environment, which is very cool. But yeah, I also don't have a great answer for that. Sorry. Yeah.
Do you expect to see more hybridization across all types of phylogenies as global change increases?
The question was, do we expect to see more hybridization as global change increases? I think all else being equal, yes. You know, there's several review papers written about, you know, habitat homogenization does more to, you know, sort of bring species together that would otherwise be, you know, discontinuous and promotes hybridization. The thing is that while Pizzly bears get a lot of like press, I don't think that is what is going to cause the main driver of extinction. So I do think there's probably some component of, you know, previous reproductive barriers that might be homogenized via anthropogenic change. But I generally think that's a much smaller concern than a lot of the other issues that are facing species, because you have to think about, you know, all the species, then all the species that could hybridize, all the species that do hybridize, all the species that hybridize where it would be, you know, a fitness detriment. Like that's already a small sliver of species. But it's a great question. So yeah. Yeah, so that has to do with, you know, over time. Oh, sorry. The question was why it wasn't sort of why it was kind of gene specific, this discordance in the phylogeny, as opposed to sort of genome wide, or why both genes didn't come along. And I mean, it comes back a lot to recombination. I think that these introgression events happened a long time in the past. And so, yes, that if we go back to this little schematic that I put up, so in this case, you have a whole chromosome that came from the red species into the blue species. But over time, they start getting mixed up. And if there's no selective benefit, if there's no adaptive benefit for that gene, it will, you know, evolve via what we call genetic drift. And in some cases, if it's being swamped out, but, you know, these individuals are occurring in otherwise a blue population, those blue markers, those blue parts of their chromosomes will become dominant. If, however, there's a little sliver, say that little sliver is around a gene like BCO2, that's actually beneficial. it will rise in frequency. It's just like a new mutation. So it has everything to do with recombination, the fact that you actually get this breaking up and mixing, which is how you can get it so sort of, in this case, gene specific. And it's associated with the time since hybridization, right? The first event is basically the whole chromosome. Then it's bigger chunks and it's smaller chunks. And people actually use the size of the region of what they think moved between the species as an indicator for when hybridization happened. We haven't done that in the system, but it's a very it's something that we could measure. Yeah, absolutely.
Thank you so much, Dave. What, Mark?
This is a kind of weird question.
Oh, I'm into that.
There are genes that are designed to introgress.
Well, I wouldn't use the word design, but I would use the word, do they have a higher propensity to, or do they have characteristics that might, so I don't have an answer, but what I will suggest, and I think it's completely anecdotal at this point, but probably the best, so this, I've told you about introgression between closely related warblers, right? The best example of the next level, like introgression on steroids, is what we call horizontal gene transfer. So it's between organisms that are in different phyla, different kingdoms. The best example, the cover of Science example of a gene moving between, in this case, aphids and fungi, right, is a carotenoid processing gene. And it's happened multiple times, primarily in herbivorous insects. So spider mites, aphids, they've acquired these carotenoid processing genes, presumably from the plants that they are ingesting. I don't understand how the mechanism of, there's some mistake in the DNA repair machinery that they incorporate these plant genes into an aphid. So if you do a phylogeny of aphids and you use just that one, like the co-phylogeny there is off the charts, right? Because it looks like it's a plant, but it's just part of their genome. But what are the chances that like the coolest introgressed gene in these warblers is a carotenoid processing gene? I mean, there is like, that's a case where plants have clearly solved the problem of generating a biochemical pathway that can process these complex molecules, whereas birds and other animals have not. And so maybe that could be considered a propensity, but that's like an N of 2. So, but I think I don't think there's anything necessarily about the genes or the chromosomal position or their characteristics that would make them more or less likely. I think it has more to do with how big a benefit does that function present. And in that, I mean, in the aphid to herbivore, they're actually consuming the animal or the plant and the DNA that could then potentially be incorporated. But it's well, I want to say it's very rare, but people have found a handful of examples. But it's a fun philosophical question. And I mean, we're interested in asking whether these introgressed genes facilitate a higher level process, which is like the process of speciation. I mean, can you predict speciation from like gene sequences? I mean, I'm not suggesting we can, and I don't think we will be able to, but it's worth asking. So that was a little bit philosophical. Oh, I'm sorry, but
Thanks so much, Dave.
thanks, Sas. Thank you. Oh, sorry. I'm sorry.
How much data have you collected as part of the project and how do you store it?
Oh, so we, terabytes, and we make it freely available online as soon as we publish. So that means it's stored on the NCBI repository so that you can go to a link and you can, if you're excited about this, you can just download all the data. But because, transferring has become a pain. Like I literally just sent a terabyte USB drive to LSU because it's just too cumbersome to transfer. But yeah, so we triply back up everything for the grad students and people working with big data in the audience, because we've had disasters. But yeah, it's a lot of data that we, a lot of it's all on the computing cluster. So both cloud and on hard drive. Thank you. All right.